Hey everyone, welcome back. Today, we're doing something a little different. We're zooming in, way in, on something that's literally right in front of us all day, every day, but completely hidden from the naked eye. Yep, we're talking about the skin. It's our largest organ, our main shield against the outside world, and honestly, it's like this incredible canvas that tells the microscopic story of our genetics and our environment. But to actually read that story, whether it's a simple itch or a complex skin cancer, we have to connect what we see on the outside with what's going on down at this cellular level. So I am super excited to walk you through this visual explainer today.
Here's the roadmap for our journey today. First, we'll establish the baseline. Then we'll look at irritated skin, followed by the broken barrier. After that, we'll tackle benign growths, and finally, malignant transformations.
All right, section 1, the baseline. Because we really can't spot the chaos until we know what normal looks like, right?
Let's dive right into this. Look at how incredibly orderly a normal epidermis is. It's like a perfectly constructed brick wall. Down at the very bottom, you've got the basal cell layer, or stratum germinativum. These cells act like the foundation, and they're constantly dividing. As we move up, we hit the prickle cell layer, then the granular layer, and finally, right at the top, the horny layer, the stratum corneum. And get this, that top layer is made entirely of dead, flat, nucleus-free keratinocytes. It's literally a shield of dead cells keeping us safe. And underneath the whole shebang sits the dermis, which is packed with blood vessels and hair follicles. Really burn this beautiful, orderly image into your brain, because things are about to get messy. Okay, before we go looking for trouble, we need to quickly grab the right vocabulary. One term you're going to hear me throw around is acanthosis. It's just a fancy way of saying a thickening of the epidermis. Basically, the cells in the middle layers, the stratum malpighi, start multiplying like crazy and cause the skin to thicken. Nailing down this term right now is going to make reading these upcoming pathology slides so much easier.
Let's move on to section 2, the irritated skin. So here's that acanthosis we just talked about live in action. This is chronic dermatitis, which you probably know better as eczema. Look at those downward finger-like projections of the epidermis, the papillae. See how broad and stretched out they become compared to our normal baseline? You can also spot an inflammatory infiltrate hanging out in the dermis underneath. It's essentially the skin's classic response to chronic scratching and irritation. It physically thickens up just to protect itself.
Now, viral infections also hijack our baseline structure, and honestly, the way they do it is fascinating. This right here is a verruca, basically your common wart, caused by the human papillomavirus. Notice those elongated ridges? They're actually pointing inward toward the center of the lesion, almost like they're trying to cup it. And if we were to zoom in even closer on those upper layers, we'd see what are called koilocytes. These are specific, almost bubbly-looking cells with a clear halo around the nucleus. When a pathologist sees that halo, it's basically a direct fingerprint left behind by the virus.
Now for section 3, the broken barrier. Things are taking a bit of a destructive turn. Blisters, medically we call them bullae or vesicles, but the crazy thing is they really aren't all the same. The absolute secret to diagnosing them is figuring out exactly where that skin barrier broke. This diagram beautifully maps out the possibilities. A blister can form high up in the epidermis or right above the basal layer, or it can happen entirely below the epidermis, literally lifting the whole top layer of skin right off the dermis. So finding the anatomical location of the split tells the pathologist exactly which disease they're dealing with. And this comparison just brilliantly illustrates that concept in real life. On the left, we're looking at Pemphigus vulgaris. Check out how the blister forms right above that bottom layer of cells. That's a suprabasal split. It leaves this single row of basal cells attached to the dermis, and pathologists actually say it looks like a row of tombstones. Kind of grim, but highly memorable. Now compare that to Bullett's Pemphigoid on the right. Here, the split is way deeper. It's subepidermal. The entire epidermis is lifted clean off the dermis. So to the patient, both just look like a blister, but under the microscope, literally two entirely different diseases.
Let's shift to scaling conditions, because they also have very distinct microscopic signatures. This one is psoriasis. What's happening here is the skin cell turnover is on hyperdrive. It's moving so fast that the top cells don't even have time to shed their nuclei, which is a condition called parakeratosis. But the real hallmark? Guide your eyes up to the very top layer. See those tiny dark purple clusters trapped up there in the pink horny layer? Those are called Monro microabscesses, essentially tiny collections of white blood cells. If you spot those, boom, you've confirmed psoriasis.
Another major scaling condition is lichen planus, and its textbook criteria are super interesting. It's got hyperkeratosis, sure, but what really gives it away visually is the interface right between the epidermis and the dermis. The ret ridges take on this incredibly sharp, jagged, saw-toothed appearance, and right beneath those saw teeth, there's this dense, band-like infiltrate of inflammatory cells that just hugs the basal layer, literally attacking the junction. It is a highly destructive but visually unmistakable process.
All right, section 4, benign growths. This is where we start connecting what the patient sees in the mirror with what the pathologist sees under the lens. On the left is a macroscopic view of a squamous cell papilloma, basically a fleshy, stalk-like skin tag. Now, on the right, under the microscope, look at how that stalk is actually built. It's got a core of fibrovascular tissue, so blood vessels and connective tissue, and it's completely surrounded by a massive overgrowth of those normal epidermal cells we talked about back at the baseline. It's just normal skin architecture that decided to grow outward.
And here's an incredibly common benign lesion, especially in older adults: Seborrheic keratosis. The big takeaway here is at the lateral margin, the very edge of the lesion. Look at that sharp demarcation. It drops off in this almost perfectly straight line right back into normal epidermis. That crisp boundary is a classic, super reassuring sign that a growth is benign. You might also spot those swirling circular structures inside the lesion. Those are called horn cysts. They're just filled with keratin and are totally characteristic of this completely harmless condition.
But all this brings us to a pretty critical question, doesn't it? We've seen skin thicken up to protect itself, we've seen it blister under an autoimmune attack, and we've seen it form these harmless, fleshy little growths. So, when does a normal cell actually cross the line and become a dangerous, invasive threat?
Which leads us to our final section, Section 5, Malignant Transformations.
What's absolutely wild about cancer cells is how they desperately try to remember what they were originally supposed to do. On the left, you're looking at a gross, fungating mass of squamous cell carcinoma. It looks chaotic. But on the right, in the microscopic view, look at those swirling masses of bright pink material right in the center of the dark tumor cells. Those are called horn pearls. These invasive, malignant cells have pushed way deep down into the dermis where they absolutely do not belong. But they are still trying to produce keratin, exactly like those normal top-layer skin cells do. It's this completely disorganized, dangerous attempt at a normal bodily function.
Then we have another huge player, basal cell carcinoma. Clinically speaking, this one is deeply tied to sun exposure. As you can see, it almost exclusively pops up on hairy skin, most commonly on the face, specifically above an imaginary line drawn from the earlobe down to the corner of the mouth. It usually shows up as a slow-growing little nodule with a central ulceration. In fact, historically, doctors called it a rodent ulcer because it quite literally destroys local tissue by burrowing down into it.
Let's see exactly how that rodent ulcer behaves under the microscope. Look at those large, solid nests of dark purple cells invading down into the deep dermis. Now look really closely at the very edge of those nests. The outer cells are lined up parallel to each other, almost exactly like a picket fence. We call this a palisaded appearance. Also, do you notice those stark white gaps right around the tumor nests? That's actually a shrinkage artifact. The tumor cells physically pull away from the surrounding tissue when the slide is being prepared in the lab. That clefting, combined with the palisading, are total dead giveaways for basal cell carcinoma. Okay, this brings us to what is easily the most practical, need-to-know info for literally anyone who has skin. It's the ABCD rule, and it's how we compare a benign mole to a malignant melanoma. A benign mole is symmetrical, it has a smooth, well-demarcated border, uniform color, and it's small, usually less than 6 millimeters. A melanoma, on the other hand, breaks all those rules. It shows asymmetry, border irregularity, color variation, all within the same spot, and a diameter greater than 6 millimeters.
Let's put that all together and see the complete picture. On the left is the gross specimen of a melanoma. Notice the incredibly erratic borders and that chaotic mix of black, brown, and tan. It completely fails the ABCD test. And on the right, the microscope shows us exactly why. That normal cellular order we talked about at the start, it's entirely gone. There's intense junctional activity at the border, heavy, totally irregular melanin pigmentation, and most concerning of all, atypical mitotic figures. That means the pathologist caught the cells right in the act of rapid, uncontrolled division. The chaos we see on the surface of the skin is literally just a reflection of the profound chaos happening at the cellular level. So,
What's the big takeaway from all this? It's that our skin is so much more than just a wrapper holding us together. From the beautiful, precise layering of a healthy epidermis, down to the sawtooth ridges of inflammation, all the way to the erratic borders of a melanoma, every single bump, blister, and freckle is a window into a vast, incredibly complex cellular universe. Our skin is quite literally telling the microscopic story of our genetics and our environment. So, the next time you notice a weird freckle or a scratch on your arm, I want you to ask yourself, what exactly does that look like under the microscope right now? Keep observing, keep asking questions, and I'll see you in the next explainer.